I want to show you another way to think about what's going on in mitosis and meiosis. So let's draw an XY axis here. The y-axis is amount of DNA, and so we have to figure out some way to measure that. So for this example, let's just say number of base pairs. And I'll say DNA. So number of DNA base pairs. We're not going to actually put numbers on that axis, but to give us some unit there. And down here we're going to have G1, S, G2, and then M for mitosis. Okay, so if we have a certain number of base pairs, we start in a particular cell. In a human, it would be, who knows, it'd be a lot, millions and millions of base pairs. But we have a certain number of base pairs. By base pairs, by the way, I mean in DNA, you have an A here, so the other strand of DNA would have a T, and here you have a G, the other strand of DNA would have a C, so on and so forth. These are base pairs. So how many base pairs are there? So we'd start here and we would double that in S phase because we copy the DNA, and then when we get to mitosis, we would go back to our original value. So this is how much DNA is there in each cell. That cell doubled its DNA and then divided into two cells, so now each cell has the same amount of DNA they started with. Now if we look at meiosis, we're going to do the same thing. This will be G1, S, G2, M1 for meiosis 1, and M2 for meiosis 2. So here we start off with the same amount of DNA we did up here. The DNA duplicates. We have twice as much now. Then we come back down. Then we come back down again. So in meiosis, we actually produce 4 cells. Here we had 2 produced. 4 cells, and each of these cells here have half as much DNA as we started with here. We replicated it. We divided it up into 2 different cells here, but remember in this case, chromosomes were still consisting of sister chromatids stuck together. Then we separated those out in meiosis 2, and we end up with 4 cells produced, 4 gametes in the case of humans, 4 spores if you're a plant, but 4 cells are produced that have half as much DNA as we started with. To see that in a column graph form, maybe it'll be clearer for you. Here I have amount of DNA, like the proportion of DNA in an organism, 1 being 100%. We start off with the cell at 100% in G1, and G2, in S phase, that cell duplicates its DNA, so it has twice as much as it normally does, and that's still the case in G2. And then in mitosis, things split out into 2 different cells, so each cell has as much DNA as we started with per cell. In the case of meiosis, we start off at 100% of what a cell should have in terms of amount of DNA. That DNA duplicates in S phase, and in G2 phase it's still duplicated. In M1, meiosis 1, at the end of that phase we have 2 cells produced that have about the same amount of DNA as in G1. It's in a different configuration than it was before in G1, but it's about the same amount. And then in meiosis 2, that number halves again, so we have half as much DNA at the end of meiosis 2 than we had when we started this process. Okay, so if that doesn't make sense, think about it, draw it, work on it until it does, because if you understand this, then you're really starting to understand what's happening during mitosis. This is just how much DNA is in each cell, right? And then you can start wrapping your head around crossing over and independent assortment and all those things that mix up variability. But this is about going from diploid to haploid, or in the case of mitosis, going from diploid to 2 diploid cells being produced.